Hello, we are team number 1 and we are going to present genetic engineering in food production. The importance of optimal nutrition for human health and development is an important concern for humans. Adverse environmental conditions such as drought, flooding, extreme heat, and so on affect crop yields more than pests and diseases. Thus, a major goal of plant scientists is to find ways to maintain high productivity under stress, as well as developing crops with enhanced nutritional value. Genetically modified crops can prove to be powerful complements to those produced by conventional methods for meeting the worldwide demand for quality foods. Crops developed by genetic engineering can not only be used to enhance yields and nutritional quality, but also for increased tolerance to various biotic and abiotic stresses. Integrations of modern biotechnology with conventional agricultural practices in a sustainable manner can fulfill the goal of attaining food security for present as well as future generations. Food insecurity and malnutrition are currently among the most serious concerns for human health, causing the loss of countless lives in developing countries, and agricultural biotechnology is proving to be a powerful complement to conventional methods for meeting worldwide demand for quality food. With the help of modern plant biotechnological tools, today we have access to massive gene tools that can be exploited to impart desirable traits in economically important crops. Plant biotechnology has the potential to address various problems in agriculture and society. Genetic modified strategies are being employed to minimize yield losses due to various stress, both biotic and abiotic. Insect resistance, Bt crops, and or herbicide tolerant genetic modified crops, which are currently under commercial cultivation, have benefited farmers through better insect and weed management, higher yields, and reduced chemical pesticide use. Sustainable integration of conventional agricultural practices with modern biotechnology can enable the achievement of food security for present and future generations. However, it is important that the performance of a genetic modified crop is closely scrutinized for several generations under field conditions and goes through rigorous biosafety assessments on case-by-case basis before being released for commercial cultivation. Because at the end of the day, genetic modified crops are going to be an essential part of our life, and the enormous potential of biotechnology must be exploited to the benefit of humankind. Among all the possible benefits of genetic engineering, we can find more nutritious foods, less use of pesticides, increased supply of food with reduced cost and longer shelf life, faster growing plants and animals, food with more desirable traits. On the other hand, some people have expressed concern about genetic engineering foods, such as creation of foods that can cause an allergic or toxic reaction, unexpected or harmful genetic changes, and foods that are less nutritious. Just in 26 countries, GMOs were grown, mostly in small farms. Among them, 10 countries were from Latin America, and as we can see in the map, in 75 countries, GMOs were grown, imported, or used in field trials. Here, we can appreciate how the GMO crops have been adopted in the USA, and we can see an important growth during the last 25 years in the percentage of farmland per crop. This reflects how genetic engineering in food production have developed through the last decade, and the trust on this type of technologies has reached an important level, at the point that the most adopted GMOs is the herbicide-tolerant soybean. Also, we need to take into account that genetic modified crops are going to be an essential part of our life, and the enormous potential of biotechnology must be exploited to the benefit of humankind. Now, we are going to present a video that will expose several points regarding the use of genetic engineering in food production.
I'll just call it an aberration. It's that Frankenfish is what we call it.
Frankenfish.
That Frankenfish that they're talking about? Well, this is it. It's a bioengineered fish, and it's got a lot of people feeling a lot of things.
It is so unnatural.
It's the first bioengineered animal ever approved for consumption, and after 30 years of debate about whether we should eat it, it's now on its way to your dinner table. And it may forever change how we produce food. We're here outside of Aqua Bounty's Indiana facility. Just last week, they received their first shipment of Aqua Advantage salmon eggs. Now these eggs, when harvested, will become the first genetically modified animal ever approved for consumption in the US. So these are the eggs. They don't look like much, but they're the culmination of decades of research and debate over whether genetically modified animals have a place in our diet.
We're going to see a 2 billion person increase in the population in the next 30 years. So we're going to have to feed 28% more people. So you start with that premise, and then you think, how is seafood going to play a role? Wild-caught salmon is basically at full capacity. Other farmed method is ocean cages, and they have their own set of challenges because those fish are exposed to climatic conditions, predators, disease.
And that's where Aqua Bounty's fish come into play. 30 years ago, scientists figured out how to produce a salmon that grows to market weight in half the time as conventional salmon. To do this, they injected the growth hormone from a Chinook salmon into an Atlantic salmon, along with a hormone from the ocean pout to ensure the rapid growth is constant. These super fast growing salmon have two big advantages. First is that they need a lot less food.
They have what we call an improved feed conversion ratio. And what that means is for every pound of feed that you put in, you get more than a pound of meat out. So if you think about cattle, it takes about 8 pounds of feed to create 1 pound of meat. With our fish, you put a pound in and you're getting like 1¼ pounds out.
Since they grow faster, Aqua Advantage fish require 25% less food than conventional salmon. The second advantage of fast-growing salmon is that you can simply produce a lot more of them, and that's what unlocks Aqua Bounty's other upside. They grow their fish in what boils down to big tanks, but the facility is actually pretty high-tech and has some huge upsides. Super tight controls on the conditions prevents disease, nearly all of the water is recirculated so it's ultra efficient, and you can put them pretty much anywhere. The problem has been that it's so expensive. But when you have fast-growing fish in there, the numbers start to balance out in a way they never have before. All of this may sound great, but it's been far from a cakewalk. Genetically engineered anything is rife with controversy. The long road to market is the result of an approval process that has surfaced two consistent and pretty reasonable questions that arise when considering genetically modified animals. What if they get out and overrun native populations? And more prominently, are we sure they're safe to eat? At the Indiana facility, Pete, the farm manager, is the point man for making sure the first one remains moot.
We have, first of all, on a really large scale, we have geographical location. We're in Indiana. There are no native salmonids. Nevertheless, inside our units, we have a very high level of physical containment. Every possible escape route out of the facility, water discharge points, pipes, they have layers of containment, physical barriers in place that will prevent the passage of the fish. And we do have numerous redundant layers.
And on top of the physical barriers, nearly every single fish is sterile. If one gets out, it can't breed. So then the question remains, is it really safe to eat? After decades of asking the same question, the answer from regulatory bodies is yes. Why did it take that long? Well, since there was nothing else really like it, there was no process for it.
So in the case of animals, they didn't have a really good law, so they used the Federal Food, Drug, and Cosmetics Act.
This is Dr. Eric Hallerman from Virginia Tech. He was brought in as an expert during Aqua Bounty's approval process.
What they said is the transgene is basically a drug because it's altering the structure or function of an animal. In a traditional food like Atlantic salmon, it's not regulated at all for that. In fact, we learned a lot about the biochemical composition of Atlantic salmon because Aqua Bounty had to go and do baseline research on what is conventional Atlantic salmon. Then they had to show rigorously that theirs was no different.
And ultimately, that's exactly what the FDA decided.
Basically, the way that FDA pithily put it, Aqua Bounty salmon is Atlantic salmon. That's it.
With all the regulatory questions settled, Aqua Bounty has the green light to sell their salmon on U.S. shores. And now the looming question is, what does this mean for the industry? Does Aqua Bounty's long fight with regulators clear the path for more innovations like it? Or does it serve as a warning to anyone else trying to bring bioengineered animals to market? Dr. Allison Van Eenennaam from UC Davis fears it's the latter.
I've worked in this field for, almost 30 years, and there's one product that's ever got to market. I mean, how depressing is that? Because there's been a number of really useful applications that have been developed over the last 20 years, and they're all basically sitting on the shelf because no one has been able to raise the capital to bring them through the regulatory process. But the problem is that it precludes the use of that technology in food production and, more concerningly, in developing countries. The average first world consumer has no idea that their choices are actually kind of impacting the ability to use innovation in the developing world. I always find it strange to be categorically opposed to technology. I think you have to look at it on a case-by-case basis, and I think most people want healthy and safe food and also to reduce the environmental footprint of our existence on Earth. And quite honestly, I can't think of an industry that more requires, innovative technology to reduce the environmental footprint of food production.
Conventional methods are not going to feed 2 billion extra people on this planet. And it certainly is going to happen without some pretty significant impact to the environment. We're proud of the fact that, we solved a problem and we use science to do it.
As seen in the video, there are controversies on the use of genetic engineering on food industry. But if the world population is expected to grow as much as 2 billion in the next 30 years, we need solutions. And one of these solutions is the use of genetic engineering in food industry, like in the 7 applications we are going to discuss next. The first application we are going to discuss today is the flavor saver tomato. This was the first crop to be genetically modified and sold to the market back in 1994. The main characteristic of this tomato is that it can have a longer shelf life due to the suppression of an enzyme called polygalacturonase, or PG. When the product was launched to the market 27 years ago, it was a commercial success. Nevertheless, in 1998, its sales began to decline, mainly due to the fact that a British biochemist told in television that some rats have been presenting several biological effects due to the consumption of a genetically modified potato, not because of the product of the introduced gene, but because of the genetic engineering process itself. Thus, concerns were raised on the whole genetic engineering processes in the food industry. Despite these affirmations being proven to be false, the sales of the flavor saver tomatoes were so low that they were no longer sold after early 1999. In the flavor saver tomato, the protein that wants to be suppressed is the PG, who has been found responsible for the dissolve of the cell wall pectin in the tomato, which is the reason why the tomato softens as it ripens. The genetic modification performed in the tomato was insertion of a reverse orientation copy of the gene, with the aim of suppressing or at least diminishing as much as possible the expression of the PG. Also, in order to be able to create the PG antisense tomato line, it is necessary the presence of a cannabis-resistant gene as a selectable marker. As mentioned before, the PG gene was isolated from the tomato and inserted by reverse orientation in order to avoid expression of the protein. And all this process was done by agrobacterium-mediated transformation. The plasmid used for this objective was PCGN 1416, which is the one that we can see here in the image. We can observe several important biobricks. For example, we can see that the plasmid has a T region, since the process is an agrobacterium-mediated transformation. As well, we can see the cannabis-resistant gene, and most importantly, the pgcDNA in the antisense orientation. Now, according to Colgene, which is the company that developed Flavor Saver tomato, the vegetable is almost indistinguishable among non-genetically engineered tomatoes, except for 2 main characteristics. The first is that since PG enzyme is being suppressed, the tomato has a longer shelf life due to the fact that the cell wall degrades slower than those of non-GMO tomatoes, and that the paste, which was canned and sold massively back between 1994 and 1999, was observed to be more viscous when compared to traditional tomatoes. And now here in this image, we can see a comparison between non-GMO tomatoes and the Flavor Saver tomato. As we can see, the differences are quite impressive. As early as 20 days, we can see that the traditional tomato has already wrinkled a lot, and by 45 days, it is in an inedible state. But if we see that the Flavor Saver tomato, we can see that neither in 20 nor 45 days, the tomato looks in bad condition. Now we're going to talk about another application. This application is the transgenic virus-resistant papaya. This is produced in the United States, in the state of Hawaii. This state is one of the main producers of papaya, especially a region called Puna. 29 years ago, in 1992, it was discovered that a virus that has been affecting the crops of papaya. And this virus was the papaya ring spot virus, Prsv. In this application, the pathogen-derived resistance was used, meaning that the resistance is obtained when the genetic material of the pathogen itself. The process of resistance was done with biolistics by inserting a code protein of a mutant of Prsv in embryogenic cultures of a type of papaya called sunset. And here in the image, we can see how the virus affects the papaya, giving it some rings on its peel. And as an important result of the genetic modification is that the resistance to the Prsv is durable, at least in Hawaii. Due to this, it was necessary to set up experiments that show if the papaya is resistant to only the Hawaiian strain of Prsv or also to other strains of Prsv. The experiment showed that the papaya was resistant to the Hawaiian Prsv strain, but susceptible to the Prsv strains with origin in Asian countries such as Taiwan, Thailand, or Guam. Also, in further studies, it was found that the pathogen-derived resistance is caused by the presence of interference RNA, which silences some genes. And we can see here in the image a healthy papaya crop in Hawaii, which were more commonly seen starting 1999, because from 1992 to 1998, the Prsv was severely affecting the papaya crops all over Hawaii. And while since the genetic modification of the papaya lies on the resistance of a virus, and viruses, as we know, mutate frequently, it is necessary to monitor if the rainbow papaya crops still have the resistance to Prsv. And the genetic modification of the papaya crops helped to recover the production of papaya in the region of Puna in Hawaii. In fact, in 1998, when the virus was in its peak, the production of papaya was only of 26 million pounds. And 3 years later, when the resistance crops were already present, the production of papaya rose up to 40 million pounds. And well, as we can see here in this table, the production of papaya in 1992, the year the virus was first identified, was of 53 million pounds, only in Puna. And the two subsequent years, 1993 and 1994, the production increased, having a peak of 55.5 million pounds in 1994. But in the following 5 years, a severe decrease was observed, having the lowest production in the year 1999 of only 25.6 million pounds of papaya, not even half of the production of 1994. In 2000, 2 years after the introduction of the transgenic seeds, the production started to grow again, not recovering to the levels present at the beginning of the 90s, but it was possible to have a peak production of 40.3 million pounds of papaya in the year 2001. And due to these reasons, many experts agree that the biotechnology and genetic engineering have played a key role in the production of papaya. And it may be possible that if genetic modification of papaya was not done, the papaya will not exist nowadays, or it will be very difficult to produce due to its susceptibility of the virus.
Now, I'm going to talk about the genetic modified salmon. The development of a genetically modified salmon is one of the advances in genetic engineering within the past 30 years. The Aqua vintage salmon founder animal was generated in the 1989 by microengineering a recombinant deoxyribonucleic acid rDNA construct composed of an element from an ocean food antifreeze protein gene and a protein coding sequence from a Chinook salmon growth hormone gene into the fertilized egg of wild Atlantic salmon. This salmon, produced by Aqua Bounty Technologies, is a triploid, effectively sterile, female fish containing a rDNA construct called OPA-FP GHC2, which imparts a rapid growth phenotype, allowing populations of these animals to reach a common growth measure, a small size, more quickly than populations of comparator Atlantic salmon. This new food animal engineer was developed by combining DNA from three animals: the Atlantic salmon, the Salmon salar, deep-water ocean elput, Macrosoarces americanus, and Pacific Chinook salmon, Oncorhynchus sadwisha. It is important to mention that Pacific Chinook salmon are a different species from Atlantic salmon. In order to manufacture an animal that grows unnaturally fast, the genetic engineering causes overproduction of an insulin-like growth factor hormone. Therefore, the new animal purportedly grows to commercial size in half the time of conventional Atlantic salmon. As shown in the image, the growth hormone from Chinook salmon joins a promoter from an ocean put, an eel-put fish spliced into an Atlantic salmon DNA, and finally, the new growth hormone directs the gene to produce hormone all year round instead of only in the summer. Now, you may be wondering if genetic engineered salmon are not manufactured to be sterile. Well, the answer is yes. The genetic engineered salmon eggs are treated to be sterile, but these methods are only 95 to 97% effective. With hundreds of thousands of eggs in production, these inevitable failure rates result in thousands of fertile fish. The adult salmon that have been engineered to fertilize these eggs, broodstock, are not sterile and could mate with native salmon in the wild. On the other hand, it is important to mention that if genetic engineered salmon escape into the environment, they will threaten native species via predation or competition for limited food or space, transgenic contamination, and an increase in exotic disease and parasites. Once engineered organisms are let loose into the environment, it is impossible to recall or eliminate them. Unlike chemical pollution, GA contamination is a living pollution that can propagate itself over space and time via gene flow. Now, I will explain some of the pros and cons of the genetically modified salmon. Starting with pros. Genetic engineered salmon could help meet rising demand for fish. This genetic modified salmon eats 25% less food, thus makes fish farming more profitable. And finally, it could reduce pressure on wild fish stocks. And now, on the other side, the cons are that fish could escape from farms and harm wild salmon populations. Science on long-term effects of genetic engineered salmon is limited, and the concerns about genetic modified fish could further upset the ocean's delicate ecology, causing an ecological disruption or other species extinction. Now, the image of the right shows both fish at 18 months of age. It can be seen that Aqua Advantage salmon can reach adult size in the 16 to 28 months instead of 35 months for regular Atlantic salmon. This transgenic salmon eat 25% less feed and are about 20% more efficient converting the food to flesh. Now, I'm going to talk about the alfalfa. USDA first deregulated herbicide-tolerant alfalfa in 2005. Alfalfa, a perennial herbaceous forage crop known as lucerne, extensively grown in the world, is utilized as hay, silage, and for pasture. It is valuable feed for many classes of livestock because of a large dry biomass yield, high protein, minerals, and vitamins content. Due to its ability for nitrogen fixation, alfalfa enriches the soil, reduces the need of fertilization, and supports the growth of other plants. Improvement of some genetic characters, like resistance to abiotic and biotic stress, protein quality, and digestibility, could significantly increase the yield and nutritional value of alfalfa. Agrobacterium-mediated gene transfer has been basically used for transformation of embryogenic alfalfa genotypes. Transfer alfalfa plants were obtained by particle bombardment of kali and pollen. Efficient transformation was also achieved through microinjection and electroporation. Now, talking about its herbicide resistance, it is interesting that most of the trials carried out with alfalfa are focused on resistance to herbicides and diseases, thus resulting that weed problems are one of the most serious and difficult to control. One of the most destructive weeds in many alfalfa seed fields is dodder, Cuscuta spp, an annual parasitic weed, which causes 20% reduction of forage yield and blocks the seed production. Genes conferring resistance to herbicides were among the first which had been identified and used for this purpose. The first of alpha transgenic plants expressing a gene of practical interest was the one called BAR, that encoded resistance to the herbicides glufosinate ammonium, were obtained by Agrobacterium TI plasmid mediated transformation and analyzed under greenhouse and field conditions. And in order to promote alfalfa herbicide tolerance, the processes of Agrobacterium tumefaciens mediated transformation were optimized. Two different bacterial strains. The first one is the LBA4404 and GB3101, containing respectively binary vectors carrying BAR gene for contact non-selective herbicide BASTA and AHAS3R gene for selective herbicide spleen were used in these transformations. It results interesting how, like other crops, transgenic alfalfa can be also used as bioreactor for producing of industrial enzymes, an approach significantly cheaper and conventional in contrast to expenses for constructing new fermentation facilities. In the following chart, we can appreciate how the alfalfa herbicide tolerance has increased in recent years to the genetically modified plant.
One of the most important applications of genetic engineering in food is the golden rice. It is well known that rice is the main staple food for hundreds of millions of people. This product is generally consumed without the outer layers, pericarp, tegument, etc. And the main reason is to remove the oil-rich iron layer, which turns rancid on storage. As the result of this, the edible part of rice grains consists of the endosperm, filled with the starch granules and protein sporate, causing the lacking in several essential nutrients for the maintenance of health. Therefore, dependence on rice as the main staple food contributes to vitamin A deficiency in at least 26 countries in the world. Golden rice was designed to produce beta-carotene, which is a natural precursor of vitamin A in the edible part of the rice. The original plant is capable to produce beta-carotenes in the leaf, where the precursor is involved in the function of photosynthesis, but it does not produce beta-carotenes natural in the endosperm because it doesn't require the use of photosynthesis. This is due to the absence of three enzymes, which are phytotin synthase, phytotin desaturase, and lycopene cyclase. Initially, the scientists tried to introduce all these genes into immature rice embryos, step by step, individually by particle bombardment. But it didn't work very well, and therefore Agrobacterium mediated transformation of immature rice embryos was used, designed to install to complete B-carotene pathway into the rice endosperm in a single transformation effort. Three vectors were constructed. The first one is the PB19HPC, which combines the sequences for a plant phytotin synthase originating from Narcissus phaedonarcissus and the sequence coding for a bacterial phytosin desaturase originating from Erwinia erudobora. These two being placed under the control of the endosperm-specific lutein. The second one was carrying the same enzyme as the first one, but with a lacking of the expression cassette for the selectable marker APH4. And the last one was carrying the sequence coding for the enzyme lycopene cyclase under gluten promoter control. Bananas resistant to Fusarium. Fusarium oxyporum is a fungus that causes a disease called Panama disease, which affects the roots of a banana plant. The spore of this fungus infects plant roots and colonize the xylem vessels, blocking the flow of water and nutrients, causing extensive necrosis and the plant death. The problematic is that the spore of this fungus lie dormant in the soil until a substitute banana plant is established. Banana crops also present a lack of genetic diversity due to their asexual propagation, make them super vulnerable to disease. Also, this fungus can be eliminated from soil by any chemical treatment, also present resistant to any fungicides, and there is no treatment to cure this disease. The solution was the genetic engineer research from the Queensland University of Technology in Australia are working in a project where the Cavendish variety was modified with the addition of RGI2 gene, obtaining a variety called grand name Cavendish resistant to TR4. The first gene, CED9 anti-apoptosis gene derived from the nematode Canorhabditis elegans, confers resistance to fox race 1 in transgenic ladyfingers bananas. The second gene, RGA2, isolate resistant gene analogous to, is a putative nucleotide binding and leucine-rich gene from a seedling of Musa acuminata, which resistant to TR4. They constructed both CAD9 and RGA2 derived transgene expression cassette, where CAD9 was under the control of the mice polybiquitin promoter and RGA2 was under the control of the Agrobacterium nopalin synthase promoter. These cassettes were transformed separately into Cavendish cultivar grand name, making it resistant to this disease.
So now with genetically modified corn, corn is the most commonly grown crop, and most of its GMOs, corn is created to resist insect pests or tolerate herbicides. Bacillus thuringiensis BT corn is a GMO corn that produces protein that are toxic to certain insect pests, but not to human, pets, livestock, or other animals. These are the same types of proteins that organic farmers use to control insect pests, and they do not harm other beneficial insects such as ladybugs. GMO BT corn reduces the need for spraying insecticides while still preventing insect damage. While a lot of GMO corn goes into processed foods and drinks, most of it is used to feed livestock like cows and poultry like chickens. Corn is one of humankind's earliest innovations. It was domesticated 10,000 years ago when humans learned to cross-pollinate plants and slowly turned a scratchily nondescript grass called teosinte into plump, productive modern corn. Genetically modified crops have the fastest adoption rate of the new technology in global agriculture simply because farmers benefit directly from higher yields and lower production costs. Here are some benefits and risks. Benefits. BT crops were intended to help farmers reduce not only the impact of pests, but also the use of agrochemicals in modern crop productions. According to Leonard Gianesi, Senior Research Associate at the National Center of Food and Agriculture Policy, farmers who adopt GM crops make more money in tougher times. Indeed, insect and virus-resistant traits have already saved several industries. According to the recent International Council for Science, ICSU, review of GM crops, disease-resistant corn crops may have lower levels of mycotoxins, potentially carcinogenic compounds to humans. They result from fungal activity in insect-infested corn crops. With fewer insect holes in plant tissue, associated fungi are not able to invade and produce toxins. Here are the risks. As pollen and seeds move in the environment, they can transmit genetic traits to nearby crops or wild relatives. Many self-pollination crops, such as wheat, barley, and potatoes, have a lot of frequency of gene flow, but the more promiscuous, such as sugar beets and corns, merit great concern. Seeds are more likely to spread GM's sugar beets genes into wild relatives. And breed so intensely for hundreds of years, most crops cannot survive without human intervention. Bt corn is a type of genetically modified organisms. Corn produced through biotechnology is being used in many familiar foods, including cornmeal and tortilla chips. Corn is used to make high fructose corn syrup, which is used as a sweetener in many foods, such as soft drinks and baked goods. The FDA, U.S. Food and Drug Administration, considers Bt corn to be nutritionally equivalent to the traditional corn. To transform a plant into a GMO plant, the gene that produces a genetic trait of interest is identified and separated from the rest of the genetic material from a donor organism. In the case of Bt corn, the donor organism is a naturally occurring soil bacterium, Bacillus thuringiensis, and the gene of interest produces a protein that kills Lepidoptera larvae, in particular European corn borer. This protein is called the Bt delta endotoxin. Growers use Bt corn as an alternative to spraying insecticides. So here's the vectors ZMIR13L. This was used in event MON863 corn, which is resistant to coleopteran. This vector is necessary for the Bacillus thuringiensis CRY3BB1 gene, which is the one that prevents the pest in the corns. As a conclusion, the whole issue of genetic modification and their use for food is a sensitive area and one that should be handled carefully with as much public debate and discussion as possible. If the technology is to succeed, then its application must be treated as honestly and transparently as possible to avoid the mistake incurred with food irradiation. Genetic engineering is beneficial in aspects associated with global hunger, food production, and pesticides reduction. We prepared a Socrative activity for you. You have until Tuesday, the 25th of May at 6 p.m. to answer the quiz. You have unlimited time. Please put your full name, and any technical issues, please contact Javier at his email address or any of the team members. Thank you very much.